We have a talk today by Dr. Mahmoud Bukar Maina. Sir, I hope I'm pronouncing your name correctly. Please correct me if I'm wrong.
It's all right. Thank you.
Thank you so much, sir. Dr. Maina is a research fellow in Sussex Neuroscience at the University of Sussex. His research focuses on understanding the basic mechanism of disease in neurodegenerative diseases, with a focus on Alzheimer's, as you guessed. Dr. Maina is also passionate about promoting a public understanding of science and inspiring and training young scientists in Africa, and now exclusively at NeuroNovember with us. He has published numerous articles and has organized workshops to train neuroscientists. And he has also founded the Trend in Africa Outreach Branch in 2013 and serves as the outreach coordinator, as well as the Science Communication Hub Nigeria and the African Science Literary Network. For his extremely important work, he has received numerous awards, including the Royal Society of Biology Science Communication Award in 2017. And in 2020, the year of all years, he was endorsed by the Royal Society as an exceptionally promising scientist. Over to you, sir. We're honored to have you speak with us.
Thank you very much, Shani, for the introduction. I'm really excited to be here. Let me try to share my screen so that um, everyone will have access to it. All right. So let's see. Can you see my screen?
Yes, sir, we can.
Yeah, all right, cool. So thank you very much again for the introduction. I'm really, really excited to be here. And it's really exciting to see that the audience is quite broad, from adults in high school to early career researchers and established scientists. This is really, really exciting. I'll try my best to kind of make the talk not boring to the high school students, but at the same time, not too boring to the early career researchers. But again, thank you very much to the organizers for having me. Well, here in Sussex, it's around 11 o'clock or 11:30, and I can see in India it's about 5 o'clock. So that's really, really, really kind of strange and exciting. All right, so let's start. So as you've heard from the introduction, I work on Alzheimer's disease, but I guess it's really important to start by talking about dementia itself. So there's a group of diseases that we kind of, that are termed under the umbrella term of dementia, and these diseases are kind of have one similar symptom, which is that there's an irreversible degeneration or brain cell death in these brains of people with these diseases. And because unlike other tissues, such as say liver or heart, etc., where cells die and you can see some level of regeneration, in the brain, most of it does not go through that process. Of course, there are other areas that are involved with brain cell kind of born again, and but in terms of neurodegeneration, such as in dementia, that really doesn't happen. And that is really, really worrying. What is really worrying about this is that the number of cases of people living with dementia is in the rise. So for example, if you look at this figure, you will see that in Americas, by 2050, it's suspected that about 30 million people would have the disease. And in Africa, where I was born and raised, the incidence rate is quite low, but you will see that it is going to rise up to 16 million or 15.8 million if we are to be precise. And in Asia, the number is rising. So this is really, really a problematic situation, simply because the population is also aging, and age is one of the risk factor of dementia. When we say risk factor, it means that as people age, because their basic kind of biology is also kind of changing, they become more susceptible to developing dementia, but it doesn't mean that they would get dementia. All right, so moving on to the real talk today, I thought that I should show you some of the figures, some of the kind of people who are celebrities that have died or have suffered from dementia. So for example, Ronald Reagan, the 40th US president, had Alzheimer's disease, and I think it contributed to his eventual death. And Robin Williams, you probably know him, he's a popular actor. He died in 2014, and he was later found to have died from Lewy body dementia. And Sir Thomas St. Connery, who recently died, it was found that before he died, he was suffering from dementia. So actually, not just the celebrities, there are a lot of people that have had dementia, people that are not known who are going through this difficult disease. And unlike other diseases, one problem with dementia is that it takes away your memories even before death. So you could say that someone who has cancer, even if it is a debilitating cancer, and he is expected to die from it, he has his memory and he can see his loved ones, he can remember all these different things. But with dementia, especially for example Alzheimer's disease, which is one type of dementia, people lose their memory. So even before they die, they kind of lose connection with the world, they kind of forget who they are and what is of value to them. So this is one of the problematic things with dementia, because it makes you stop existing even though you are alive. All right, so now moving on, as you have heard, my focus of the talk would be on Alzheimer's disease. And Alzheimer's disease was actually first described by Alois Alzheimer's in 1906. But of course, you know that this disease has been existing before Alois Alzheimer's time. But he was the first person to describe it based on the case of a patient called Auguste Dieter. And like you've heard, some of the symptoms of Auguste Dieter kind of featured included memory loss, hallucinations, etc. And when Alzheimer's performed a pathological evaluation, histopathological evaluation of her brains after her death, he found that within her brain, first, there is a reduction in the brain size, obviously, or let's say brain volume. And within her brain, if you take a chunk of it and you look at it, he found some two interesting clumps of proteins. So he found that there is something called now amyloid plaques, which are found outside the brain cells. And then another thing that now is called tau tangles, which are found inside brain cells. So one thing that is really important to know about Alzheimer's is that even though now we are finding new tools to be able to diagnose whether someone has Alzheimer's or not, but one of the ways to prove that someone had Alzheimer's is to do a histopathological analysis on their brains after their death. So these people, they die after their death, they donate their brain, and they do this histopathological analysis. And if they see presence of amyloid plaques, these ones, and tau tangles, then they say, oh, this person confirmed died with this disease. But to really understand what I'm going to be talking about today, it's probably important to understand the A beta generation, what it is and how it is generated. So don't be don't take this slide too serious. There's nothing complex about it. The main thing that I'm trying to say in this slide is that A beta, which is the one that forms these amyloid plaques that clump together outside brain cells, is produced through the action of two enzymes. So this is a cell. You can see this is a cytoplasm and this is the extracellular space. And in the membrane, there is a protein called amyloid precursor protein. And as the name suggests, amyloid precursor protein is the precursor protein for A beta, for amyloid beta. So first, there's an enzyme called beta secretase. When it cleaves this amyloid precursor protein, that is when it cuts it just like a scissors would cut down something, it leaves a fragment, this fragment here, and another fragment here. So you don't really need to know the name, just know that there are two fragments. And after this cleavage, there's another enzyme called gamma secretase. When it comes and cleaves this fragment here, it now leads to the production of amyloid beta, A beta here, and another fragment that is called C59. So this is called the amylogenic pathway. But actually, if a different enzyme called gamma alpha secretase started cleaving this APP, and then gamma secretase comes to cleave it, A beta is not produced. So you only get A beta when gamma secretase cleaves or kind of cuts this APP here. So this is how it's produced. But what is important and why I wanted to show you this is because I wanted to really show you that there are a lot of mutations that have been found in different families around the world, which leads to the development of Alzheimer's disease. So for example, this is the beta secretase side where it cleaves the protein. And for those that are in high school, I need to remind you, or maybe you've heard from your biology classes that um, in the body or in our cells, we've got the genetic makeup, which is inside the nucleus. And this genetic makeup is made up of a DNA. And then the DNA now goes on to form the RNA, and the RNA then goes on to form the protein. So this kind of proteins, or this RNA and DNA, they are in the form of sequences. So here, this DAE that you see, they represent in amino acids. And it is the RNA that gives this sequence or this information to produce this protein based on a combination of these different amino acids. So all these alphabets that you are seeing, they represent a particular name of amino acid. And what was really found was that there are a lot of mutations on this APP and also on the enzymes that produces it. Some of these are called Swedish mutation, liver mutation. So depending on the name, you'll know where it was discovered or on which families it was discovered. So French mutation, German mutation, all these different mutations actually lead to a change in the production of amyloid beta. And therefore, they result in the development of Alzheimer's disease, a specific type of Alzheimer's disease called familial Alzheimer's disease. And it's called familial because it is developed through genetic inheritance, through the inheritance of these mutations. And in around 1992 and 91, a lot of work was done, showing that the mutations can cause Alzheimer's, and this led to a hypothesis called amyloid cascade hypothesis. So remember, Alois Alzheimer's reported Alzheimer's in 1906, but it took nearly 70 or 80 years before a significant progress was made. And this led to this amyloid cascade hypothesis. And in this hypothesis, it said that in the form of Alzheimer's disease that are inherited, that is the familial Alzheimer's disease, mutations on APP or presenilin 1 or 2, which are kind of the genes that go on to produce these enzymes that help in coating the amyloid beta or APP. So the mutations in this lead to increased production of amyloid beta 42. And if you go back to this slide, you will see that you could end up getting a lot of different A beta, either A beta 38, 40, 43, 48, 50, etc. So this A beta 42 particularly has been shown to be the most notorious. Once there is more A beta 42, it is associated with more cell death or brain cell death. And for other forms of Alzheimer's disease, which are not through inheritance, they are usually called sporadic AD. They said that the failure to clear A beta from the brain can lead to increase in A beta 42 levels. So in both this and this, what is happening is that you have increase in A beta. And then they go on to say that this increase in A beta will lead to its oligomerization. What oligomerization means is that they kind of stick together, they bind to one another to form bigger kind of chunk of protein. So this can lead to a problem in the synapse. So these synapses are kind of like connections between brain cells, and this can lead to gradual deposition of this A beta into these plaques that I showed you, which deposit outside brain cells. And this can also lead to microglial and astrocytic activation in the brain, which can result in inflammatory responses. This can lead to oxidative injury and lots of different things, which can result in dementia. So basically, based on this amyloid cascade hypothesis, they say that amyloid beta is at the top of the kind of food chain, if you want to call it. So it's at the top. It is what causes all these different things that lead to the development of the disease. So, and lots of research has shown us actually that when you have A beta, it goes on to stick together or kind of forms these clumps, and these clumps actually go on to form these amyloid plaques. And it starts by this monomers. So monomers, just you say a single kind of A beta peptide, and this aggregates to form diamers like two, and then this goes on to form oligomers, and this goes on to form protofibrils, etc. So a lot of research has shown that this process actually happens. And again, in about 20 years ago, or kind of close to that, it was found that oligomers of this A beta are actually very toxic. And based on that, it was suggested that actually in Alzheimer's disease, it is these oligomers that are responsible for the brain cell death that happens in the disease. So this is just to show you, this is a model from a group in the United States that actually tried to mimic how these proteins stick together. And you can see that they are kind of like small ones come to fuse with another small one, and this leads to the formation of this big chunk of kind of aggregates. So it's kind of cool to see this because it kind of gives you an idea about how these proteins aggregate or kind of clump together to form these plaques. So now going on to what we do, it's important to know that oftentimes when you have a problem, a human problem, you don't really ask people that have that disease to come, let me cut you a piece of brain and see what happens, because that's not ethical. So because of that, you have to have a model of studying that problem. Yeah. So in this case, for example, oftentimes you can start an experiment from test tube, then go on to brain cells in a dish. So these are brain cells, but you're growing them outside a human body in a dish. And oftentimes you go on to use a mice or rat or whatever model. And then oftentimes you then go on to look at whether that changes that you see in these different models are found in the human brain. And in this situation, it can be that you go to the brain bank to get brains that have been donated by people who had the disease before they died, or it is actually sometimes you can get these brains from after surgery. For example, people that have epilepsy, etc., oftentimes they have these surgeries to remove those parts that are causing that problem. So you can collect that brain and try to do some experiments on it. And for us particularly in our lab, we oftentimes use these preparations from test tube, and then we add them to brain cells in a dish to see what happens. And oftentimes we also use animal models to try to see whether things are similar from what we see in the brain cells in a dish. And sometimes then we also get this brain tissue to try to extract A beta or to try to see what happens with collaborators. So in the last 10 years, since a lot of work has been published from our lab, some in which I have participated and some from my colleagues, showing how amyloid beta could be important in driving disease, or in in the early changes that we see, which could be important in Alzheimer's disease. But for this talk, I'll focus more around protein synthesis, and therefore it is important for me to give you a small background about this process. So, in molecular biology, maybe in high school, you've learned this as well. And there's something called the central dogma. And the central dogma is basically a kind of dogma that shows that what really happens in our cells is that first we have the DNA, which is like the cookbook having all the instructions. This DNA now goes on to produce an RNA, which is like a particular chunk of information from that DNA, which goes on to form proteins. Yeah. So it is now these proteins that end up to do a lot of different things in our body. And so it's really important to know that the process of protein synthesis is essential for the growth and survival of all living organisms. It's really crucial. If your cells are not producing proteins, you're gonna die. I mean, you need proteins, you need it to do all the different things that is important in your body. But then it's also important to know that these proteins that are produced, there is actually something that is happening before they are produced. And there is a particular kind of structure inside the cell called nucleus. And inside this nucleus, there is another structure called the nucleolus. So this is the nucleolus here, yeah? And this nucleolus is important for producing the building blocks for making ribosomes. So think about it. If you want to cook a food, you want to have the recipe, you want to have the ingredients, but you need the pot. The pot would be like the wood. You can't cook without a pot, you know. So ribosomes are more like the pots, if you know what I mean, because they help in allowing you to put all your ingredients and the recipe and everything so that you can cook it. So without a pot or without a ribosome, you can't really cook. So the nucleolus is important for producing the building blocks that are important for this ribosome formation. And if you go a little bit deeper, this is the nucleolus. And what really happens is that there is a particular form of DNA called the ribosomal DNA, which goes on to produce the ribosomal RNA. Sorry, I'm using these technical terms, but it's kind of important. So this ribosomal RNA go on to produce these kind of building blocks that go on to form the ribosomes, which go on to help with protein synthesis. So what I really want you to know is that the nucleolus has a function that is necessary for life, because for proteins to be produced, you need the ribosomes, and the ribosomes need the building blocks which are produced by the nucleolus, yeah? So a lot of work has shown that in Alzheimer's disease, the ribosome dysfunction is an early event. And a lot of work has also shown based on histopathological examination that the protein synthesis machinery or building blocks is damaged in Alzheimer's disease. Yeah. So now, based on this, I've done some work in the last 2 or 3 years ago, in which we wanted to really understand what is the early process that happens in Alzheimer's, but using brain cells in a dish as a model. And since oligomers are believed to be the important or drivers of this toxicity, we wanted to find out if you prepare oligomers in a test tube and you add them to brain cells in a dish, what really happens. So a lot of work in our lab has shown, like I shown you earlier, has shown that these oligomers can go on to kill cells. So for example, this is a work from my colleague, Karen Marshall, which actually is 2020, not 2016, which shows that when you treat cells with these oligomers, either 1 micromolar, 5 micromolar, or 10 micromolar, it goes on to cause the cells to begin to die. So this shows you that when they are incubated, these are primary neurons. So these are neurons that have been grown from rat. And when they have been treated with amyloid beta, after 14 days, they died, mode cell death, and then 7 days, less than 14, and 3 days, even lesser. After 24 hours, you see a really small percentage of cell death. So this green dot shows you the cells that have died. So really, my question was, what are the earliest damages caused by amyloid beta in neuronal cells? So before you begin to see the cell death, what really is happening before that time? Because that will be key for us to understand or to find therapeutic targets. Now, one of the first few experiments that I did was, again, I prepared amyloid beta in a test tube, and then I added them to brain cells in a dish. And I allowed them to remain for about 24 hours, after which then I decided to investigate what is happening. So this is an assay called cell viability, using cell tableau. Basically, with this assay, you'll be able to see whether your cells are healthy and alive, they are viable. So we did this assay, and you can see that after 24 hours, there is no cell death, there is no significant loss of cell viability. And then going forward, there is a particular kind of understanding in the field that one of the process that happens before cell dies in most situations is that there is DNA damage. So here I try to look at whether there is DNA damage in these cells using a marker called gamma H2AX and fluorescence microscope. And again, here you see that there is no DNA damage. However, very interestingly, I found that there's oxidative stress. So oxidative stress is more like accumulation of free radicals inside our body, and usually these free radicals, we need to reduce them. There are processes that help in reducing them. If they are not, that can result in oxidative stress, and that can lead to brain cell death or cell death. So you see, there is not DNA damage or loss of cell viability, but we have oxidative stress. So now it's important to go back to the nucleolus, because it has been shown that the nucleolus serve as a stress sensor. And given that it serves as a stress sensor, one of the things that I wanted to do was that, since I've shown there is no cell death, but we have oxidative stress, I wanted to find out whether the nucleolus is affected. So now I said, what happens to the nucleolus following amyloid beta administration? And here, whenever I say amyloid beta here, I'm talking about amyloid beta 42. So now this is a kind of confocal microscope image or double staining with a nucleolar marker. This red, which is FBL, it stands for fibrillin. And this is a nucleolar protein. And NP tau, the green dot, is actually for a protein called tau. So when you do this double labeling, you can see them localized within the blue area, which is the nucleus. And when you then stress cells with amyloid beta, you can try to see what really happens. But I've mentioned tau here, so maybe it's important for me to take a step back and tell you something a little bit about it. If you remember in Alzheimer's disease, I told you that amyloid plaques and tau tangles are two of the main hallmarks. So, but this tau protein that you are seeing, actually, lots of work has shown that it can be found in different areas of the cell. So it's not just found in the axons, or it can be found within the nucleus, nucleolus. And in fact, we published a work showing that tau can actually be found within the nucleolus, where it plays a role in transcription and stress response. So, but the focus of today's talk is not about that. I just wanted to highlight that tau can be found within the nucleolus. My work has shown that it's important in nucleolar stress. So now let's go back to that. So you remember, this is the cells that I have treated with amyloid beta 42 for 24 hours. If you look at it, you will see this. You see these orange arrows, depending on how you see it on your screen. But these arrows here, you can see they are showing some red dots. These red dots are no longer yellow. Normally, where there is yellow, it means that there is co-localization, or it means that these green and red proteins are in the same place. However, here, when you have amyloid beta treatment, you can see that there are some spots that are only red. And this tells you that the green protein is not inside the red protein anymore. So this is one of the features that happens when there is a condition called nucleolar stress. So when the nucleolus is under stress, some of the proteins begin to become redistributed. And actually, you can quantify that. You can quantify how much of this green is inside this red area, and signal that is lost inside the green area, inside the red area. So this tells us that fibrillin, which is that red protein, and nucleolar tau, the green protein, are redistributed following A-beta administration for 24 hours. So now, you remember the central dogma of molecular biology, DNA to RNA, and then RNA to proteins. So now, another key thing about the nucleolus is that when it is under stress, some of the proteins that are found in the nucleolus also become degraded. So I wanted to now find out, does the amyloid beta administration affect these proteins? Now, this is a Western blotting, and with this, this TIP5 is a protein found in the nucleolus, and UBF is a protein also found within the nucleolus. It's a transcription factor, and fibrillin. You can see that the amyloid beta administration leads to a reduction in UBF and TIP5, although at this time point, the fibrillin is not affected to that much extent. So what this tells us is that some of the nucleolar proteins are being degraded, yeah? And again, going back to the central dogma of molecular biology, RNA comes from DNA before the protein. So here in this experiment, I did qPCR, which is a technique that can allow you to measure the concentration of RNA in cells. I looked at ribosomal RNA. And if you remember ribosomal RNA from the name, It is an RNA that is important for ribosome formation. So here you can see after amyloid beta administration for 24 hours, all the different forms of ribosomal RNA, 18S ribosomal RNA, 20S ribosomal RNA, which come from the 45S ribosomal RNA, are all decreased. So this tells us that A beta leads to a reduction in ribosomal RNA. Yeah. So, but it's important to know a reduction in ribosomal RNA will result in a reduction in ribosome production and eventually protein synthesis reduction. Yeah, because if you remember, this is a nucleolus, it produces the ribosomal RNA, which goes on to form these building blocks that go on to form the ribosomes. So based on that logic, I said, okay, what happens to protein synthesis? Is there any changes in protein synthesis? So here I did a particular assay called click-ITR ribosomal protein synthesis assay, which tells us how much proteins are synthesized in the cell. And you can see with this assay, I found that the amyloid beta treatment for 24 hours led to reduction in protein synthesis. But there are many different ways, different things that can lead to reduction in protein synthesis. And one of these is a phosphorylation of a protein called EF2 alpha. Sorry for the high school students here, but just think about it as a pathway that can lead to reduction in protein synthesis. So I checked whether this is affected, and I found that it is not affected by the amyloid beta treatment. So this now tells me that the reduction in the protein synthesis is actually because of the reduction in ribosomal RNA, not because of the phosphorylation of this protein here. All right, so now it's important to find out what happens first, because I'm trying to understand what are the early changes that amyloid beta causes in the body or in cells, which could be important for triggering Alzheimer's or for driving Alzheimer's. So in this experiment, what I did was, in last experiment, I treated the cells for 24 hours, but here I treated the cells for 2 hours so that I can see what happens early on. And what happens actually is that the cells are viable, they are not dead, there is no DNA damage, but still even at 2 hours, there is oxidative stress. And then when I looked at the nucleolar proteins, just like I showed you earlier, I found that UBF, that other protein that I showed you earlier, which was decreased by the amyloid beta for 2 treatment for 24 hours. So actually UBF is decreased, but not T5 and not fibrillin. And also there is no change in protein synthesis. So what this tells us is that at 2 hours of treatment, you have small increase in oxidative stress and subtle nucleolar stress or subtle changes in the nucleolus. So this now led me to conclude that the early consequence of A beta treatment in cells are oxidative stress and subtle nucleolar stress, which over time becomes worse, leading to a reduction in ribosomal RNA levels and eventually a decrease in protein synthesis. And if you look at it, earlier on, I showed you that there are a lot of papers showing that in Alzheimer's disease, there is a decrease in protein synthesis, there is a problem with the ribosome. But actually, this work in a brain cells in a dish model showed us that the culprit is actually amyloid beta 42. All right, now to conclude, to go towards the conclusion of my talk, we know that A beta is toxic, but what is the driver of this toxicity? Yeah. So for example, we know that there are different forms of A beta. The monomers go on to aggregate to form oligomers and eventually amyloid plaques. And we know that oligomers are toxic. And some researchers have shown that protofibrils are also toxic. So this is a paper that we recently published, and full credit to Devki, who really did this work. And what we showed is that these oligomers, when you make them in a test tube, and you also make fibrils, and these fibrils you can see they are like webs or like long fibrils, or when you take these fibrils and you sonicate them so that you have broken them down to smaller bits, and you treat primary neurons with them, only the oligomers go on to kill the cells. The fibrils and fibrils that have been sonicated do not kill the cells. So what this tells us is that oligomers are toxic, protofibrils based on work from a different group of people have been shown to be toxic, but the fibrils are not toxic. And if you remember, this is how they form. Small come to bind to a bigger one, and then this go on to form these amyloid plaques that we see here. So really, in our lab, we think that what really drives toxicity is this process of assembling together rather than this particular species of oligomer, protofibril, or fibril. We think that it is this process that you are seeing in this movie, this kind of things coming together to bind to one another, that is really driving the toxicity. Because think about it, if you have a house and then you bring in a machine that self-replicates, that keeps kind of growing, it will grow to an extent that your whole house will be full. So we think that it is that process that really causes this toxicity. But how can we address that question? So the best way to address this is to see when you are having this growth, kind of freeze them in time. So you are seeing this growth, then you freeze them in time so that you can freeze oligomers, protofibrils, fibrils, and you treat this into cells to see whether they are toxic. So this is exactly what I've addressed recently. And I did that by using another, a process of cross-linking called the tyrosine cross-linking. So the tyrosine cross-linking is a cross-linking mechanism in which when a tyrosine in a protein is close to another tyrosine, they can bind together to form the tyrosine. And this is quite stable. And this is the sequence of the A beta that we use. If you see the sequence, this green here is Y, which is for tyrosine. So which means we can actually use the tyrosine cross-linking to stabilize amyloid beta from this assembly process to see whether whether it is the driver of toxicity. But in any experiment, and this I'm saying to the high school students, in any work that you do, you need to have a control that can show you that what you are doing is not as a result of something different. So here we have another A beta called the variant A beta, which we have shown in the lab, which has been shown in our lab is not aggregating. It doesn't bind to one another. While the first one, A beta 42, it binds to one another, forming these plaques. So in this work, this is the work that I've done in the test tube. And when you mix amyloid beta with copper, you can oxidize it. Or when you mix A beta with copper and hydrogen peroxide, you can even create more oxidation on the A beta. So this is exactly what we did for the A beta and for the variant A beta, which is called VAB here. And following that, you can then use fluorometer to see whether you have dityrosine, because you can dityrosine has this fluorescence that is intrinsic. So when you excite it, it will show you a signal like this, which shows you that dityrosine has been formed. So you can see here the samples that have incubated in a test tube with A beta and copper and hydrogen peroxide, they quickly form this dityrosine, which is here in yellow. And even the one that has been the variant A beta here also forms dityrosine. Yeah. Now, one thing that I want you to really focus on here is that you see at 2 hours, you see this blue line, it begins to form dityrosine. And that is the one that has been treated with copper. So what we can really do is that we can follow the formation of these assemblies or this kind of A beta binding to one another. We can follow that using an assay called THS binding assay. And that can tell us whether there is binding of A beta to one another to form these fibrils. So what we see is that the A beta that has not been oxidized, which is this one, it goes on to form fibrils. And the A beta that has been incubated with copper, which is this one, remember it begins to form at around 2 hours. And exactly around that 2 hours, once there is the tyrosine formation, it becomes stabilized. It doesn't go on to form fibrils like these ones. And all the other ones with the hydrogen peroxide, it doesn't even go on to form fibrils. So what we went on to do is to do an electron microscopic examination. And with this, we can see that the A beta goes on to form these fibrils. You can see like webs here, while the A beta that has been incubated with copper or with copper chloride, they don't go on to form these fibrils. They have been stabilized. And the variant A beta, we know that it doesn't assemble, so nothing happens there, so it's a control. So what this tells us is that the diatyrosine cross-linking is stabilizing the A beta. But then there's a problem here. Because we are adding copper and we know that copper can influence amyloid beta aggregation. So we really need to find a situation where we do not add copper so that we can rule out any problem that copper is causing to the peptide. So we then did another experiment, and in the experiment we used UV to cause the cross-linking of A beta, that is photooxidation. And still using that, you can see that diatyrosine is forming here. And what we then went on to do is to look at the fibril formation. And you can again see that the A beta that has been oxidized with the UV, it doesn't go on to form these fibrils. And with the electron microscopy, you can see that it also doesn't go on to form these fibrils, which A beta forms after 5 days. So what this tells us is that when you use UV to photooxidize A beta to form this diatyrosine cross-linking, it also stabilizes it, similar to when you use copper. So now what we then decided to do is that since now we can use diatyrosine cross-linking to stabilize either oligomers or protofibrils or fibrils or whatever we want, we can stabilize them in time when they are aggregating. We now decided to stabilize them at different time points with the diatyrosine cross-linking, and then look at whether they will go on to kill cells. So now in this experiment, we stabilize them using this oxidation to cross-link with A beta, and then at the different time points, we treated neuronal cells, and then using high content imaging that allows you to kind of measure and maybe changes in thousands of cells, we now looked at the toxicity. And what really is exciting here is that this orange is A beta. You can see that at 2 hours after treatment, 2 hours after making A beta, and then you treat the cells, then after 3 days of treating the cells, when you look at them, they kill cells more than when you have A beta that has been oxidized to form this diatyrosine cross-linking. So when you have this A beta that has been stabilized, it doesn't kill cells after 3 days, but the A beta that has been made for 2 hours, but which has not been stabilized, meaning that it is still growing, binding to one another, after 3 days, it goes on to kill cells. And when you do the same, you make A beta, you allow it to sit for 24 hours, during which you know that you have some protofibrils, and you stabilize it with the diatyrosine cross-linking. The ones that have not been stabilized go on to kill cells, but not the ones that have been stabilized. And at 48 hours again, if, for example, you make the A beta, we know that at 2 hours they are oligomers, at 24 hours they are about protofibrils, at 48 hours they are like protofibrils and fibrils. When you do that and you stabilize them, you see that they do not kill cells, but the ones that have not been stabilized go on to kill cells. At 4 days, none kill cells, and we know that at 4 days, even the A beta that has not been stabilized would have formed fibrils, and the fibrils we know are not toxic. So at that 4 days, whether you stabilize them or not, they don't kill cells. So what this really tells us in conclusion is that when the protein is self-assembling, when they are binding to one another, kind of forming these clumps, that is what gives them the tendency to kill the cells. And that is what gives them the tendency to cause that protein synthesis inhibition that I've shown you earlier. However, when you stabilize them, they lose their ability to kill cells. So we believe that the driver of toxicity, or what makes amyloid beta toxic, is this process of self-assembly. I think I've run out of time. So with this, I would like to end this talk by thanking all the people in our lab, especially my long-term mentor, Professor Sapel, and my colleagues who have contributed to some extent in some of the work I've shown you. And of course, I would like to thank my family who are there always and supporting me to continue doing this research, and the funders that have funded the work that I've been doing over these years. Happy to take questions.
Thank you so much, sir, on that talk and taking us through that journey of how amyloid beta causes oxidative stress and stress in the nucleolus to ultimately decrease protein synthesis. And as someone with little to no knowledge on this topic, I was just being able to say this much about it. Yeah, I would just like to thank you for making the most intimidating slides also seem easy to understand. And also thank you for shedding light on the importance of studying Alzheimer's, because as you said in the beginning, that with this disease, you kind of stop existing while you're still alive, and that's no way to be alive. So quite a few questions have popped in on the chat. So I'll just read them out, and we'd love it if you could answer the same.
Sure.
So Ritz had asked, what's your take on the correlation of antidepressants taken in the early age being a probable risk factor for causing Alzheimer's?
So I have to be honest with you. I don't know the answer because in our lab we do not study depression or antidepressants on Alzheimer's. And one thing that is really important to point out is that Alzheimer's disease is a very complex disease.
Mhm,
You have people working on, for example, just A beta for their entire life, just
mhm,
trying to understand how the assembly process happens. You have people working on tau protein, for example, which is the other protein that I've shown you. And you have
mhm,
people working on the risk factors. So even within the field, when we go to conferences, we end up seeing people that are working on something entirely different from what we
mhm,
do, even though they are also working on Alzheimer's disease. So it is quite a complex disease. And in science, oftentimes we just find our niche and we try to understand what really happens.
mhm.
So I don't really know any relationship between antidepressants and A beta or Alzheimer's disease development. Sorry.
No, no, thank you for your honesty. Shivani asked, is APP an essential gene? Does it have any normal function? Because if we're able to remove APP, then there will be no amyloid beta and no Alzheimer's disease.
Yeah, so that's a very interesting question. And APP is an essential gene in the sense that the different components of APP have been shown to play some roles. So, for example, I showed you earlier that we have the amylogenic pathway, which
Mhm,
begins when you have beta secretase cleaving APP and gamma secretase cleaving that kind of component leading to A beta generation. But you also have the other pathway where you have the gamma secretase first cleaving the APP before gamma secretase. That leads to production of a protein called P53 and the other component. So those other proteins have been shown to play a role, for example, in synapse and other kind of cellular functions. And even A beta itself has been proposed or has been shown to have some antioxidant property to some extent.
mhm,
So you could say that removing APP would not be like a great idea because the other components that you have within the body does have some functions. What you really want to do is probably reduce the level of A beta that is
mhm.
actually shown to be the source of the changes that we have in the disease.
Thank you. Thank you. Um, so Gayatri first wanted to thank you for sharing your work with us. I think that's seconded by everyone. She wanted to know whether there is a pore formation post-treatment with amyloid beta. If yes, then what is your opinion on A beta's role as an antimicrobial peptide?
So can you repeat the first part?
Yeah, definitely. Sorry. She would like to know whether there is a pore formation post-treatment with amyloid beta.
No, we haven't really studied that. It could be, but we haven't really studied that. And I
Mhm,
can understand maybe the reason is because amyloid beta goes on to form this mesh shock of fibers, which is similar to what happens in either pore formation or in other conditions, which are also fibril forming condition. Mhm.
mhm. Gayatri also wanted to know, could it be possible that the oxidative stress observed by a beta toxicity is a result of inflammation?
That is a very excellent question. It's an excellent question, simply because in the field there is this idea: what is the early mechanism? What is the early thing that causes this and that? And we know based on biomarker studies, for example, amyloid beta changes comes first before you see so changes in tau etc. But in terms of inflammation and oxidative stress, there is still some kind of debate into what comes early. There are some research showing that oxidative stress comes quite early in the disease, and that can, of course, trigger inflammation. But at the same time, there are studies showing that when you have inflammation, it can also lead to oxidative stress. So in reality, this is why in the field, some people are trying to focus on targeting a pathway, let's say inflammation, let's say oxidative stress, as long as it will lead to a reduction in changes that can drive the disease from progressing. So either oxidative stress or inflammation can both have a positive impact on one another.
Okay, thank you. Ankush wanted to ask, is there any way to not let the oligomers, fibrils, bind together with some kind of a mechanism like receptor blocking, something that can nullify them?
Okay, so yeah, that's an interesting question. But what I would say is that so A beta has this natural tendency to aggregate, yeah, and
Mhm.
work has been shown that A beta can bind to receptor through binding to that receptor become internalized, and work from our lab has also shown that A beta becomes internalized via clathrin dependent endocytotic pathway. So in terms of whether using a receptor to block it, I don't think that is the right approach, because the A beta comes and bind with another without the need for any receptor. They are just self-assembling. They like sticking to one another. So they don't need
Mhm.
any receptor for them to do that. Yeah. So formation of oligomer does not need any receptor. Formation of fibril does not need any receptor. It's usually through the entrance into the cell that some people have shown that receptor binding is important. But
Okay.
some people have
Mhm.
also shown that without really binding to receptor, A beta can still go on to exert its kind of damaging effects.
Thank you. Thank you. Rashi would like to ask, did the
Mm.
sex of the mouse from which neuronal cells were extracted influence the levels of A beta in vitro, as there has been proof that in humans Alzheimer's is twice as more prevalent in females than in males.
That's a very good question, and the answer is I don't really know, to be honest. I know that yes, obviously there is this in terms of developing, there is a difference between men and women developing the disease, but most of the work I've shown you are based on what we prepared in test tube, and then we go on to administer to brain cells in a dish, not on the mice model. And in our lab, we haven't really studied any difference in terms of, for example, there are people working on these mice, but the interest is not to differentiate the sex difference, but rather to
Mhm.
really look at the mechanism via which ABTA does things. And it could be that some people have shown that, but like I said, it's a very broad field, and I don't
Yeah,
really know.
yeah, definitely. Mansa would like to ask, how can we go about carrying photooxidization to stabilize amyloid beta?
Oh, well, I published the paper that I showed you earlier, or the work that I've shown you has been published. And apart from that, we've also published other works in the lab, on oxidation process. So I think the best approach would be happy to for the person to send me an email, and I can send them papers, or they can just look me up on Google Scholar. They should be able to find it.
Sure. Will your email be available on Google Scholar as well? This is just in case anybody else would like to contact you.
No, it will not be available, but it will be available on our website, on the Sussex website. So if you just check Dr. Mahmoud Maina, you should be able to find link to finding me on our website.
Okay, I'll just put that down in the chat as well.
Yeah.
In the meantime, Rashi would also like to ask, are A beta fibrils non-toxic in their actual form itself, or when they are broken down to smaller fibrils as shown by the effects of sonification?
Yeah, so that's an interesting question, and this is the work that I showed you earlier, which was published this year, which was led by Devki, and in that work, it clearly shows that the fibrils are not toxic, and even when you sonicate them to make them smaller, so that you are now having smaller size, just like the oligomers, but still they
Mhm.
are not toxic, and that goes on to support this hypothesis that we have, that it is a self-assembly, the process of binding to one another, that is important, because when
Mhm.
you sonicate the fibrils, they lose the ability to now self-assemble. So they are not toxic, and the fibrils have already matured. They do not self-assemble, they are not toxic. But the oligomers, because they still have the ability to self-assemble, they can go on to form fibrils. This is why they are toxic.
All right. Srinidhi would also like to ask, is A beta also responsible for hallucinations and patients reliving their young days?
The end of the question is not clear. Can you clarify that at the end of it?
Um, Srinidhi, could you please clarify this question? I think the main part is she wants to ask whether A beta is responsible for patients hallucinating and derailment, like straying
Oh,
away from reality.
I see, I see. So if you remember, I showed you that one of these signs that Alois Alzheimer's found based on studying Auguste Dieter in 1906, the finding that he published in 1906 in a conference, was hallucinations. She had both auditory and visual hallucinations, but that
Mhm,
came on during the late part of the disease. But what
mhm,
that really tells you is that the pathology that A
mhm,
beta causes, or the pathology that happens in Alzheimer's disease, can eventually now lead to these hallucinations that you see. And not
mhm,
just A beta, a lot of other things can cause that. But
mhm,
when it happens in Alzheimer's disease, especially familial forms of Alzheimer's disease that happens due to inheritance, then you can clearly say
mhm.
that it is due to the A beta pathology.
All right. Thank you. Thank you. Um, Mansa would like to ask, under what conditions is beta secretase produced, and can its production be prevented? Does it have any other function other than cleaving the APP component?
That is an
Cleaning?
that is
Okay.
an excellent question, and I have to be honest, I don't know the answer, and it's
Mhm,
again because the field is very broad. Oftentimes, we find our corner and we kind of try to really understand or characterize what is really happening in that corner. So, for example,
mhm,
in the last 8 years, I've been working on A beta and tau. And if I were to meet a colleague who also works on A beta or tau, but on a different things, we share learnings and we all get surprised about what our findings are because we are working on completely different things. So we
mhm,
haven't been focusing on gamma secretase, but I know that a lot of work in Bart Stroeper's lab in Belgium, who was the one of the winners of the Brain Prize, has been
mhm,
focusing on this pathway. And I'll encourage whoever is interested in that to check Bart Stroeper. And if they are interested, they can email me. I will send them the link to Bart Stroeper. He works a lot on that
mhm.
pathway.
Okay, thank you so much. I'm so sorry. I think our time is up. I've put Sir's email in the chat, so in case any of you have any more questions or follow-up questions, please do email him if that's all right. Okay,
Fantastic. That's
thank
all right.
you. Thank you so much, sir, for taking
Thank
out
you.
your time for
Thank
this
you very much.
convention.
Thank you very
Thank
much
you.
for having me. All right. Thank you.